Next, we have histone modification, which involves DNA wrapping or not wrapping around the histone. If DNA wraps itself around the histone, proteins cannot read the DNA, which turns the gene off, and vice versa. Chemical groups can add or remove themselves to reverse this change. The third change, which I have shown here on my presentation slide, involves non-coding RNA. It is slightly different as it acts more of a manager of gene expression. It does this by attaching itself to coding RNA in other proteins to break down the coding RNA until it cannot make proteins. It can also select proteins to change histones and whether it turns the gene on or off. So, at the beginning of my presentation, I showed you yeast cells, bacteria, a flower, and humans. So, but cell to cell, I want you to know that cell to cell signaling is not all that they have in common. They also experience epigenetics on different scales. According to Falk and Dolinoy, insects, rodents, and humans experience epigenetics as they face harmful chemicals and nutrients that change their gene expression. It was their environment that caused changes to their genes, which made them vulnerable to diseases. Even bacteria plays a role. As Hammond and her collaborators studied how bacteria can compromise the cells of its host, which allows it to survive longer. The bacteria, not the host. This pathogenic bacteria can even cause long-term imprints, which can silence alleles, causing a change in gene expression for the cell host. As a result, the host are vulnerable to other diseases. But epigenetics isn't new. It's been around in research since the 1900s. However, it continues to have research go back, to be as recent as 2019. And so far, I've discussed how epigenetics work in the development of organisms and in diseases. However, it does not stop there. Epigenetic changes regulate almost every cell type's function and influences intercellular communication. Under normal conditions, cardiomyocytes and endothelial cells communicate to allow cardiac function. If an epigenetic change this paracrine factor-based interaction, it would negatively affect the cardiac muscle's ability to contract. Stem cells determine if they renew themselves or differentiate by combining environmental signals with a network or transcription regulation. Regulatory modules are incredibly interconnected and heavily influence gene expression. Researchers also found that when they expose mesenchymal stem cells to nitric oxide donors with the epigenetic mixture, it caused a decrease in the expression for cardiac progenitor cells. This indicates an interruption in mesenchymal stem cells signaling pathway. The reversal of changes like this are areas of key interest for researchers. Their goals are to study illnesses, increase health improvement, and understand more about the complex pathways of cell-to-cell signaling. In conclusion, living cells, or shall I say living organisms, of all sizes and complexities, rely on cell signaling and epigenetics. It plays a significant role in health and development. Cell signaling has intracellular and extracellular molecules that allow environmental response, reproduction, and etc. Epigenetics is a phenotypic change that occurs in response to one's environment and lifestyle choices. These changes are reversible, which can cause health improvements. Epigenetics and cell signaling are connected by epigenetics by being the response to what cells communicate to each other. Researchers found these results at all levels of life as research only continues to grow. I want to thank you for exploring Trotsky, or the role of cell signaling in epigenetics, with me. These are my sources. And now I open the floor up for questions. Thank you.